Recording. Welcome everyone to the Pharmaceutical Stability Discussion Group and Stability Hub Office Hours event. We have 90 minutes of opportunity for you to bring your stability related questions, challenges, and real life experiences to your colleagues and a panel of stability subject matter experts. We'd ask that you turn your cameras on if possible, but stay muted unless you're asked to comment further on a question. Please use the Zoom chat feature to submit your questions or comments. If desired, you can address your question to a specific panel member. In the absence of your questions, we've assembled an interesting array of frequently asked questions across all stability topic areas. It's possible for us to conduct a poll on certain issue if you find that helpful. This session is being recorded and will be available in the events section at stabilityhub.com probably by the end of today. I'd like to introduce the members of today's panel in alphabetical order. First, we have Jeff Carr, pharmaceutical consultant, specializing in analytical topics as well as contract research organizations. Jeff Cook, Chief Operations Officer, Parameter Generation and Control, dealing with stability chambers, particularly those who have controlled humidity. Bashar Deeb of Precision Stability Storage, in answering questions about storage as well as auditing issues. Jeff Gardner from Data Farm, where he is a consultant there, and he's going to be dealing particularly with our statistical questions. Warren Jaworowitz, CMC Insights is his company, and he has a great deal of stability experience with therapeutic products. Maria Krish, research fellow in physical sciences from Freethink Technologies, the folks who have brought you ASAP and ISO conversion issues. Chris Latos, manager for stability services of Hollister Incorporated, deal with combination products for us. Myself, PSDG facilitator and editor at Stability Hub. Walter Routh, founder and CEO of Saguna Quality Consulting, general questions today. Ken Victor, U.S. Laboratory Manager at Lighthouse Instruments, dealing with instrumentation questions. And Sheba Zaman, who will be joining us in a while from Novatech International, dealing with software and any issues around computerization. So that's our panel. We'd like to welcome them and thank them especially for participating today. And we can open it up for questions at this point. If you have some on your chat, you can do that, or we can address some of those that I mentioned a little bit earlier. And let me pull up my chat feature. Okay, none so far, so we'll start with Jeff Carr. Question about tolerances as applied to pull dates for stability samples and timelines applied for start and completion of sample testing. So that's around what's typical, and we'd like to ask you what you've seen. You'll need to unmute for that. Still muted. There we go. Jeff's in the UK, so his sound has taken a while to reach us.
Yeah, sorry, John. I was just I thought you were waiting for somebody to jump in with some possible suggestions, but I'll just summarize the question a little bit. So for stability studies, there's a couple of timelines that are quite important to us all. So first of all, we have samples that are being pulled off according to a certain schedule, and your schedule probably in detail what dates each of the samples are supposed to be pulled off from each of the stations it's there. The second timeline that's of importance is how long do you give yourself to do the analytical testing that's required? And so it's a sort of general question, and I'll just leave it at that at the moment to see if anybody wants to jump in with some suggestions as to how we might treat these things.
Well, we've already inspired a couple of questions here. One's a kind of a takeoff on your question. It's from Denise Rin. For annual stability studies for small molecules, do you base your pull time points on the date of manufacture of the batch or on the date and chamber? And that's for small molecules or annual studies in particular, I think is very pertinent to the question. If date and chamber, do you add an expiry time point? Is that based on criteria such as the samples are placed in the chamber after X number of days, for example, 60 days? Reasoning for not using date of manufacture of the batch as the starting date of the study since samples are already stored long-term. Okay, so that's open for comment for anybody who would like to. If not, I have some opinions on that, but would be glad to yield the floor to anybody else who would like to talk about whether or not use the date of manufacturing or the date in the chamber. And you could raise your hand and unmute if you wish to comment directly. If I don't hear any voices, I'll continue with my voice. It's a big depends, I think, especially for anything that's not in the annual studies. If you're talking about annual studies, it does lean more towards date of manufacture, but there's a lot of input to that. Is it stored in a way that it's controlled and secure and access? That's part of a definition of stability studies that we had at our company. Had to have controlled access, had to be tracked and monitored temperature-wise. So that's difficult to do if it's date of manufacture, but some companies can anticipate that and do a lot of work towards controlling those conditions. So my input would be data manufacturer is okay as long as you meet those kind of criteria. And data in chamber is fine too, as long as you put in all your records that you had this elongated storage period between data manufacture and putting it in. What kind of conditions it underwent then, that kind of thing. Just as long as you're upfront with anybody that's reviewing the data and seeing that. I think you could justify it either way. So having said that, that's a big fat, it depends. Anyone else want to comment? Yes, Sri.
So the only concern on going based on the set down date and not the DOM is the way you have filed in your regulatory submission. So let's just say you filed for 36 months. In odd cases, especially in a commercial landscape, or let's just say you have 36 months of approved shelf life, your expectation is to prove stability from DOM through 36 months. And you may have batches, so your proprietary 3-batch set down, you may have one batch that went out to 38 months or 39 months based on when you have done the set down, but it doesn't amount to all the 3 batches that you need to set the 36 months. So that's where it gets a little bit convoluted to use the set down date versus the DOM. And DOM is a little more safer from that perspective. There are especially Chinese and Korean regulations. And Japan PMDA, they do come back on that to go back and do from the DOM perspective.
So lots of things to consider. Denise, I know that you've asked this question in a number of forums, and I'm hoping that you're getting to a consensus that you can present to management with some real conviction. But I don't know if we get there all the way because it is such a nebulous kind of question.
Yeah, I'll just unmute myself, John. Yeah, it's just a different philosophy at different employers. You know, how they justify it. I think part of the issue is that if you have a delay in putting this samples in the chamber, now you're looking at what does that time point represent, right? So say you take 6 months to get it on, now you're at, 36 months is really a 42. And if it fails, now you're, is it really a valid failure? If it has an expiration of 36. So I guess that's where it's really important to have the expiry time point, at the true expiry. So, but what do you do if it's post expiry, and it fails,
So I don't know if anyone wants to answer or it's just me, but But in my past life, I was at Takeda with the same kind of issues, and we made sure we added the expiry time point testing, and we did not test after. So in cases where it takes 6 months for you to do the primary packaging, the secondary packaging, and then it comes back to set down and whatnot. Yeah, we understand the delays that can happen, especially on commercial landscape. We made sure we tested at the expiry time point, and we were not responsible after that. And there were a couple of instances where the product was tested after expiry and it was out of specification, and we raised a field alert, but we basically said it was post expiry and all the drug was already distributed, so there wasn't any concern, and we never heard back otherwise. So
Okay.
Yeah, that activity there, you know, I'd say 90% of the time the post expiry data is dismissed in some way. So that's just the evidence that testing post expiry is really a huge waste of money. If you're looking for a way to save money, that would be one, to switch to your data manufacturer. A few things to consider, though, other issues involved with data manufacture is make sure your protocol, your commitment is clear on that. I've seen some protocols that say time at long-term storage, or time at 2560, for example, and the implication is this product will spend exactly 36 months at 2560, when in fact it spent only 33 months. That's one issue. The other issue is what do you do at the 3075? So a rationale for being able to wait 3 months to put it in the chamber is that the product is being maintained in its long-term condition in the warehouse or whatever. It's just being treated like normal. However, 3065, 3075 studies probably aren't if you're in zone 2. So just you might consider having an argument for why it's okay to keep it for 3 months at 2560 and then only go to 33 months at 3075. So, or don't do it. Go date in chamber for the more the higher temperatures, etc. I'll leave it at that.
Certainly an area and stability that requires a lot of finesse and justification, no matter which way you bend it. Some more expensive than others. Jeff Carr, your hand is raised. You have to unmute. Still not unmuted quite yet.
Try that.
There we go.
That should work. Okay, so the companies I've worked with, the date of manufacture is defined as the date on which the first two ingredients get mixed together. And so the batch could be a significant time before it's even been prepared. Now, the first time of testing when you do a stability study is supposed to be time 0. So your time 0 now is going to be a long time from the date of manufacture, perhaps. And that's not including the packaging aspects of it.
Okay, thank you. We have another question. It's from Brenda Clark. And we might head this in the direction of Warren if he feels like this is appropriate for his expertise. Can you give some advice on appropriate stability studies to conduct on cryopreserved cell banks? Is it sufficient to justify using routine monitoring during production as per the WHO Annex 3 TRS 978? Warren, is that in your bailiwick?
Unfortunately not. I'm more drug product than cell banks. Don't have an answer to this one.
Okay, we'll open it up for any others. I know this is an area that's the newest to stability, and we typically run short on experts in this area, but we're hoping the more people participate, the more we'll find the experts along these lines. So I can see most everybody's box on my screen, but I may have to scroll up and down a little bit or left and right to see the rest of you at the same time. Okay, not seeing anyone pop up on that one, but advice on the stability studies to conduct on cryopreserved cell banks. So these are deep frozen ones, to my understanding of this. Routine monitoring during production. So if you're following the WHO Annex, you've got some kind of guidance behind you. As far as stability goes, not much happens at that temperature, but certainly we want to offer proof of that. So routine monitoring, I think, would be okay. We're certainly open to other ideas and experiences as people have done something and then been questioned by the authorities. That's our best source to get that sorted out.
And please, if anyone else has an answer or some advice on that, we'd love to hear from you. Go ahead and unmute and speak up.
Hi, at my previous job for the production cell banks that were on stability, we looked at viability upon thaw. We had a criteria we needed to meet. And then the second thing we looked at was the ability of the cell bank to continue to produce the product. In my case, the lentiviral vector that we were making. So that's what we did for production cell banks. And then for plasmid cell banks, same thing, looking at viability, cell count, the ability of the cells to be able to be grown up again to produce the plasmid. And we did an identity test on that plasmid as well.
Okay, thank you for that input.
Was there any concern in these cryo stored cell banks about maintaining container closure integrity or sterility of the product?
So that was something that was being introduced, that sort of testing when we left, because it was something that was flagged during a regulatory audit. The container closure, the sterility, though, was easy for us to see because we would thaw the cell banks out and use them for production. And so if there was contamination, that would show up quickly during the cell expansion.
Yeah, and I can speak a little bit to the CCIT on this. Absolutely stored dry ice and CO2 ingress and storage of these products in the vapor phase rather than liquid phase of liquid nitrogen is key for contamination control and container closure integrity. You also have to look at I think the updated guidance of 382, more for drug product, but it applies here that the container closure under use conditions needs to be justified. And it's really important here because the elastomers will rebound, but in their frozen state, they become hard. So if you've got an elastomer closure, just to make sure it's properly matched with the dimensions of the container that you're using, or the threads on your cryovial are appropriate, and just make sure you're using appropriate containers for the storage conditions. Absolutely can get ingress of gas into these containers from the environment.
Right, and that's something that my company that I work with, Lighthouse Instruments, that's what we deal with. So that's why I didn't know if that was in general something that got flagged for cryogenic product, whether they were concerned about maintaining CCI in general, or if it somewhat depended on the product itself.
My experience in this has been more on the container closure than the stability programs, but we haven't done. I'm very familiar with Lighthouse and the testing on the drug product side. Never employed that on the cell bank side. It was really finding the right container closure and then ensuring, that you didn't have any problems through the functionality testing that was mentioned upon thaw, viability, production, things like that, where we looked at that. But in one case, actually, in the early days, going back to about 2013, there was an overbag, a heat sealed overbag for some of these cryo containers. So you'd actually have a secondary thermal sealed ampule type closure. It wasn't glass, but it was a plastic that would be impermeable to gas and ensure the integrity of the primary container. But in the last decade or so, the primary containers have seemed to be sufficient in my experience.
Yeah, that's so we have seen a lot of issues. We have seen issues with maintaining CCI at the cold temperatures. particularly at -80 in cryogenic storage in particular, because that is, as you're sort of alluding to, Warren, is that gets below the glass transition temperature associated with elastomers that are used to seal crimp sealed containers. And it is essentially what we helped evaluate for the COVID vaccines that were generated by Pfizer and BioNTech in the past. But that's the general idea, is that in these cold temperatures, you may lose container closure integrity only at that cold temperature. And then if you then remove that vial from the cold temperatures and then do the CCI test at room temperatures, those vials can regain integrity. So you aren't able to identify it if you're testing at room temperature that it lost container closure integrity at the cold temperatures. So the advantage of the headspace analysis is it can capture those quote unquote temporary defects that only exist at the cold temperatures. So that's something that we can help out with.
Many of these containers for cell banks are opaque. They're plastics, high density polypropylene or such, so they're not necessarily amenable to the headspace techniques. And interesting enough, in my experience, often getting these cell banks, I don't know if other people can speak to this, from research environments, the handling is quite lax, and they're often just Eppendorfs or some sort of Nalgene screw top, and they've been doing it forever, and that's how things have been developed. And then we get it in development, and we fall back a little bit and go, did you have worried about container at all? They just seem to work. So in my experience, the cryo containers, if you're buying a suitable cryo container, you've generally been okay. But I think, again, pointing to 382, USP 382, is really putting the onus more on the sponsor to confirm that the container closure is appropriate, and it's really focused on drug product. But I think the logic applies for sponsors looking at cell banks and drug substances as well. You have to make sure your whole supply lane is integral, and you don't have upset early on that could disrupt later production.
Okay. Thank you, guys. We have a question that seems to be up Jeff Gardner's alley. I don't know, Jeff, if you've been able to read it on the chat so far. It says, question on statistical analysis, 0.05 versus 0.25 significance requirement. Is it okay if we use the 0.05 significance for individual or one batch linear regression analysis? And for pooling, we use the 0.25 significance. Is it okay to just use the 0.05 significance for both individual or one batch and multi-batch pooling analysis?
All right, so let's have some fun here. Let me, in true statistician fashion, muddy the waters immediately. So the question here, short answer is yes, it's fine to differentiate between 0.05 and 0.25 as you laid it out, but let's be clear about what is being spoken about in each context. So in the case of evaluating a single batch and stability, when we're looking at a significance level of 0.05, we are evaluating the slope of that batch against a null hypothesis of no change over time, right? And so in that particular instance, it would be appropriate to use a significance value of 0.05 to test that null hypothesis. In other words, are we seeing a change in the single batch that differs significantly from a flat rate of 0% change over time? All right, so that's the first scenario. In the multi-batch scenario, when we're talking about significance, our null hypothesis is not about the rate of change over time, at least not directly. What it really is about is, are we seeing consistency among the various batch slopes in question such that there is no real difference among those batches and their slopes, or are we seeing substantial variability or difference in at least one slope, one batch slope being different from the rest, right? So the fundamental hypothesis that's being evaluated is different in that context than in the single batch context. And so in that latter context, when we're looking at multiple batches, certainly a p-value of 0.25 in alignment with the legendary ICH-Q&E guidance is appropriate in that particular instance. The more recent draft Q1 lays out also some guidelines to consider alternative models as opposed to that particular methodology, but generally speaking, yes, you would be fine using 0.25 in that instance. And then getting to the last question about, is it okay to just use 0.25 significance for both individual one batch or multi-batch pooling. Certainly for the multi-batch pooling, for the one batch scenario, is a good question, interesting question. I would say it probably is okay, but it probably would need to be justified as well. So I honestly, in my 23, 24 years of working as a statistician in a capacity, I don't know that I've ever encountered a situation where we were looking at a single batch slope and evaluated the significance of that slope using any threshold of significance other than 0.05. So this is a little bit of a unique question from that perspective, but I suppose one could do so long as you could provide the justification as to why you're selecting that higher significance level.
If I have time for a counter question,
That's okay.
Of course.
I guess. So the reason I'm asking this is we're in the process of getting a software which is validated to do all these and change the significance as I please, or as needed. But right now we're limited to JMP, and then I'm using the JMP in-build stability degradation analysis, which defaults to 0.25 significance, and it does not allow me to change that. It says that ICH asked for a 0.25, and therefore. So when I do the individual batch analysis, obviously it defaults to 0.25. So I did some digging on how to change that in the JMP, and it doesn't allow me to do it. So then I did some digging again to see, does it actually change anything when I change the significance for one batch between 0.25 and 0.5? And 0.05. And what I understood was when you do a one batch analysis, it gives you a p-value. And that p-value you measure against 0.05 significance or 0.25 significance. It doesn't necessarily change anything from a analysis perspective. So I just wanted to make sure that that assumption is actually right. And therefore I can keep using the JMP inbuilt software and be fine with it. Or I do need to find some alternative to do the 0.05 significance.
Yeah, I mean, you could go ahead and use it. I mean, the ultimate, from a practical standpoint, the implication or the consequence of using a 0.25 p-value for a single batch instance is that you're going to be more conservative in your declaration of that batch's slope as being non-zero, right? So if you use 0.05, you basically are saying, I need a pretty compelling body of evidence to declare this slope as being non-zero. If you use 0.25, you are willing to concede the point that the slope may be non-zero, even though the evidence may not be visually as compelling or obviously numerically as compelling. So that's going to be the real consequence of doing so. There's no real right or wrong, but there is the question of how conservative do you really wish to be? And, could that create indigestion that you're not really willing to, absorb, or can you live with that level of conservatism? So it's really a judgment call on the part of you and your organization, whether you could live with that.
Awesome. Thank you so much. It really helped. So.
Thank you, guys. That was enough to throw me back to some post-traumatic stress symptoms from my graduate statistics course. So let's run away over to a different bailiwick for the moment and ask Maria Krish if she can comment a little bit about predictive models on stability. Do they work across lots for different drug loads? I believe you have the question in front of you for giving us the answers on several of these points.
Yeah. Hello. Hi. My name is Maria Krish, as you said. I work at a company that does predictive models, so accelerated predictive models. Our flavor is called ASAP. And yes, it's interesting. So often these are empirical models where you're doing sort of a short-term experiment on one drug product in open conditions, and then modeling behavior on long-term stability in packaging. They are empirical, so they are really designed to look at one lot. Now, if you have different lots that are the same, expected to be the same formulation with nothing changed, you would expect that they would be the same. Sometimes these models are used to look to see if there is lot-to-lot variability that's expected or unexpected. If there are changes, they can also be used to evaluate whether those changes are significant and show up in the models. So one kind of change is a different drug load, but a common blend. From a theoretical perspective, if it's really a common blend, you would expect the same kinetics. So you would expect no difference, and you actually would expect that the models would apply. If it's a different drug load, there's almost no, very, very few cases where it would be the same. So there are a few, but in general, things change a lot with drug load. So the percent drug and a model at one drug load would not apply to others. So I think in general, common blend often matches, same formulation, different batch would often match. A comparison can be used to see if it really does, or if there's some change that wasn't captured. And different drug load probably won't match. I don't know if there's any.
Okay, any questions along those lines for Maria? Anybody have any experience using ASAP that they've been able to float with the regulatory authorities and shave some time off of their testing schedules? Okay, maybe not in this crowd, but I've experienced a number of people that have been able to do that. It's been interesting over the years to see the authorities kind of look at this with a very dubious eye to now starting to welcome some supportive data and eventually growing towards some more mainstream kind of things. So it's been really interesting to watch and see how that goes. So thank you, Maria. Let's throw a question over in the absence of a chat one over to Chris Latos about dealing with medical device stability and some of the biggest challenges you see there, what's common with regular pharmaceutical products, and what's a little bit different about the medical devices and combination products.
Yeah, for us, perhaps the biggest challenge is determining a correct stability indicating test. Unlike drug product, where when you're looking at, appearance, assay, impurities, you have some pretty well-defined stability indicating tests. Medical devices, stretch a whole gamut, anything from a tongue depressor to a pacemaker, an implantable pacemaker. Most of the medical products we make are single-use, disposable, class 1, class 2, so low-risk medical products made out of different polymers or combinations of polymers. Some of them have multiple components. So it's really trying to determine, the appropriate test to run in a stability protocol. We lean heavily on our design engineers. A lot of our testing, instead of looking at chemical attributes, we're looking at more of the physical properties. Again, these are polymers, so we're looking at how things are bonded together, and does the bond change as the product ages. So could be an adhesive bond, a heat seal, RF seal, things like that. There are some ISO tests that we use to test burst strength, or if a device is going to leak, those types of things. So for us, I think that's one of the biggest challenges, trying to determine the correct test to use in a stability protocol. Also, our samples take up quite a bit of capacity in our stability chambers, more so than drug products. I mean, we have catheters that are long, a couple of feet long, pieces of tubing, packaged and so forth. So, it takes up a lot of capacity. So it's also another challenge is trying to predict how much, stability chamber capacity we're going to need, throughout the year, and that can change. So that's kind of the biggest challenges that, I personally face.
I wanted to follow that up a bit with if you're using any particular predictive modeling for yourself, or any equations for shelf life, whether or not you can apply Arrhenius or you have the other models developed. How do you get along with an inert object when it comes to predicting? I shouldn't say totally inert, but an object that's not as viable or volatile as what we experience in the pharmaceutical world.
Yeah, so we do accelerated aging. There's a standard ASTM F1980 that we follow. Per that standard, you can accelerate age up to temperatures as high as 60 degrees C. That's assuming that you don't have any thermal transitions, so you don't have any glass transitions, or certainly you don't want your polymers melting. It's gonna wreak havoc on your stability study. So yeah, we do high temperature accelerated aging. As a contingency, we also use the standard 40 degrees C, 75% RH, just as a contingency, because sometimes, as you can imagine, at high temperature, 55 or 60 degrees C, you get some degradation that you wouldn't see at a lower temperature. Some strange things happen sometimes. So that's kind of the approach. We don't use ASAP or any type of modeling software, but we use just accelerated aging and Arrhenius model.
Okay. Does that bring up any questions for Chris in this area of medical devices or combination products? Give you just a second to respond to that.
I had a follow-up on that if I could. Chris, I don't know, on the drug product side, we often have devices and drugs together with varying expiration dates or varying manufacturing dates. Do you have any multiple component devices, and if where there are differing manufacture dates, and how do you handle that as far as testing beyond expiry and testing one not the other, et cetera?
Yeah, so you kind of cut out a little bit, but I mean, so for us, we use as the start of our stability initiation is the date that we put the product in the chamber. We don't manufacture at the site, so some of our products are manufactured overseas and sterilized overseas. So in some cases, it could be a couple of months after the date of manufacture before we receive product and put it up on stability. Per our internal procedures, we have 3 months, a 3-month target from the date of manufacture to get stability testing initiated, get samples charged in the chamber and so forth. I think your other cut out, so I couldn't hear everything, but if there's a drug constituent and a device constituent, how do you assign the shelf life for something like that? I think is what you may have mentioned. So on that one, it's going to depend on which has which one's the shelf life limiter. Is it the drug constituent or the device constituent be taken into consideration?
Thank you, Chris. One of the things I noticed in the brief period of time I was associated with medical devices, I was astounded coming out of the pharmaceutical world where something goes wrong in stability. You start looking in your batch at what ingredients were in there, if you can pinpoint something on a particular ingredient. But then I noticed when you build a device, you run out of one lot of subcomponent very quickly, and you bring in the next lot, and you might have 5 or 6 of these variations going on at the same time to produce the lot you have on stability. So I always felt challenged that if something went wrong, to try and figure out what it was. Do you much in the same way that we would take an active ingredient or components and maybe watch those on a stability study if we had suspicions, do you ever test a particular subcomponent to see if you do an acceleration on that very quickly to see if it might be the guilty party in a failure?
We tend to probably focus more on the raw materials. So again, tapes, polymeric films, things of that nature. So we do stability, shelf life studies just on the raw materials that go into our finished goods. In some limited cases, we actually do stability testing on what we call WIP. So this would be subcomponents that go into a finished device. But yeah, to your point, it can be challenging because on a finished good lot of product, you could have, you could have, 2 or 3 different, lots of one of the components, be it a film or a medical adhesive tape that go into that, just depending on the length of the production run and so forth. So yeah, it can be a challenge. Fortunately, we don't see a lot of variation from lot to lot in most of our raw materials. So.
Okay, well, thank you.
I had,
Yeah.
Oh, I'm sorry. I had one additional combination product specific question. So I'm interested just to hear maybe some perspectives on when you have an example of a combination product, maybe a auto injector or an on-body injector, where your drug really doesn't come in contact with the device components itself. Are you including like functionality testing on your ongoing annual stability program, or is that something just confirmed during process validation, and then kind of if you introduce any kind of significant change to your device, perhaps you would do additional testing? But is that like an ongoing annual commitment that folks are making?
Yeah, that's a really good question. I really don't know. I mean, we don't manufacture those types of devices like that. So I think you'd have to do some stability to at least, confirm the shelf life on the device part of it. Whether or not that's a requirement, I wouldn't think so. I wouldn't think you'd have to do an annual stability lot on the device component, just the drug component. For, strictly speaking, for medical device stability, we're not required to do, put an annual lot up on stability. Now, granted, if we make some type of change, maybe a material change, then we'll redo stability testing and so forth. But we're not required, unlike pharma, to run an annual production batches on stability.
Yeah.
It is an interesting question that brings to mind across the whole board of products. With aerosols, it'd be spray patterns and the amount of spray you get out. That's not really a direct stability thing you can analyze. It's a user kind of test. In injectables, it'd be glide force resistance, those kind of things. So I think it's an important aspect of it. It's kind of lies on the borders of the stability group, whether or not that's included in a physical test that we might run or ship it out to a different laboratory along those lines. But I think it's definitely a good side topic to run alongside with your stability data. What happened to your product? It's kind of like the counterpart of in-use testing to do that. So I'm sure that each one of us probably handles that in a different fashion, but as long as we're doing it, recording it, justifying why we test or why we don't test, even more importantly, that would be the major components of that. Any other questions along those lines? Okay, let's talk a little bit about audits. And our audit specialist for today is Bashar Deeb from Precision Stability Storage. And I wanted to ask Bashar some of the tools that might be commonly used in QA audits. I know you host audits all the time because you're a commercial facility that rely on the rest of us to send our samples there. So either all of us are there, or else the FDA has come to our shop and we point the finger over in your direction and say, hey, all the samples are over there, go see them. So can you tell us a little bit about auditing tools that you use and what kind of audits you deal with? And
Sure.
the other thing, SharePoint was mentioned in the pre-discussion as to whether or not that's helpful and is acceptable.
So that's actually one thing that we kind of started at Precision Stability Storage. So we provide storage for a lot of different things, whether it's stability or biologics. So we have all sorts of stability chambers ranging from like 2560 all the way to 4075, and then we have the -80s end. liquid nitrogen tanks. So we get clients that are basically like pharmaceutical or like hospitals or research facilities that are doing research on tissue and biologics. One of the things that we've started seeing with SharePoint is we're able to provide our clients with all of our procedures, all of our forms, all of our certification, things like that in our SharePoint prior to the audit. They do a lot of the reviews prior to that, and then we'll kind of go back and forth remotely. So it's actually helped us a lot with the auditing process to be able to get the audit done basically prior to them actually coming onto the facility. Then when they come onto the facility, they'll kind of see the facility, see what, like, you know, the warehouse areas, and then from there, we'll present them with other forms and things like that they want to actually see from our like facilities perspective on things. One of the biggest tools that we like to use, using our CAPA process here, because we want to present to our clients like, look, yes, we are very good at our jobs, but there are always like different things that could impact our facility, whether we have deviations in our monitoring systems, or we just have incorrect samples that were shipped to clients. So we try to show them that we have a CAPA process that helps us figure out what the issue is, and then eventually find out how to close out that CAPA and make sure that this issue doesn't get repeated through our effectiveness check of our CAPA process.
Sounds great. Has anybody else had experience using a SharePoint or a similar product in dealing with their contract research organizations? It's hard to send data back and forth and keep it secure, but sometimes they can put it in a place that you can go in and have access to it and others can't. And turns out to be a pretty useful way to transfer data. I've not heard of having regulatory folks having any objections to that, but I'm interested in knowing what your experience might be if you've had good or bad experiences with transferring data back and forth.
We've dealt with the Department of Health for California Biologics, and I've been sending the auditor our SharePoint links, and she's had no issues reviewing our data prior to the audit, because we're going to have an audit next week with them. And it's been pretty simple. They have the nice thing about it too is you can set timelines on it, so it'll actually expire. I like to give people normally like 30 days prior to the audit and after the audit, so that they're able to review documents before and after the audit is completed, in case they had like something that they couldn't think of, or they remembered something that they wanted to review again. And it seems to work pretty well. And you can also block them from downloading, and you can also have them be viewers, so they can just view it. And if you want to allow them to download certain folders, like our certifications folder, I'll give them viewing access, so they can actually download on that one. So it's kind of nice having those little restrictions on the SharePoint.
Sounds good. Any other comments in that area? We have a chat submission. Statistics for biologics. Anyone used Arrhenius equation to justify expiry in biologics? In the early stages, when not too much data is available, and say 2 years into the stability program where regression analysis is showing some concern. Let's see, got a little block in front of my reading material here. Some concern about the situation. How much does it matter to have data from more than one batch? We're looking to see if this strategy works for a biologic and possibly mRNA drug product. Please share any experience.
So I'll go first here. I have no experience working with Arrhenius modeling for biologics. That basically has been, I don't want to use the word taboo, but just the complexity of biological entities and the manners in which they can behave has always been communicated to me as essentially being a very, very tall wall to get over with respect to trying to model it via some sort of Arrhenius approach. But the folks at FreeThink, or this is their space, so I would defer to them in terms of any feasibility there and even success stories on successful model biologics.
Area. The firm's been trying to do a literature review of what's out there. So there are some examples. I think as the Jeff alluded to, it's more dependent. So there are some situations where it seems very appropriate and others where it's not. There have been companies that have used this strategy. There is some information out in the literature. My company, Freethink, does try and collate some of the papers that use this approach. So we have a few examples on our website. There's more in the literature. As I said, we're trying to put together actually a review paper right now, kind of collating what's out there. But there's some examples, but I think it is very system dependent. So there's some systems where it's not appropriate. There's some systems, especially if it's kind of, I think what I would say is it depends what's limiting stability. And also how tight the analytical method for testing that stability parameter is. So to do effective modeling that can give you a useful result, you typically need a method that's got no more than, say, 20% error between the difference you're looking at. Some aspects you can't get a method that tight for biologics, some you really can. So it's a depends thing, but I would say that there is a lot of interest in that area, and there's some sort of ongoing new work in it as well.
And especially when, sorry, if someone else has a.
No, go ahead.
No, go ahead, sir.
Okay, especially the thing is that from a quality stability standpoint, I have a biologic where we had a minor issue with defending expiry plus 12 months. And then the CNC folks step in and they were like, oh, we want to use the Arrhenius and we can justify this. So my argument was, it isn't typical, number one. Number two, it's a biologic. They don't follow the first order kinetics. We're into 2 years and we don't want to go that route. So that's where the question came from. And two, we are in the process of manufacturing an mRNA-based drug product very soon. And based on our trial design, we're not going to have a lot of time left. And that's why we really are trying to see if we can tap into this Arrhenius equation argument to get that expiry. So especially for the mRNA DP. So
yeah, I would weigh in here from a CMC perspective. I wouldn't be the guy telling you to use the Arrhenius plot. I'd say don't. I think it's high risk to all the comments that were said here. I think when it does match up and it appears to be predictive, it's more likely that it appears to be predictive, and in some future case it's gonna come and not be correct. These are very complex systems, and my saying is you can never have enough stability. So and especially with these products, mRNA is like, there's a lot in the literature, and there's a lot they have, especially if you're talking frozen state, even at -80, you can see changes for these products. And I'll start with the frozen. I'm really more interested in and out of condition than at condition. So what I mean by that is at -80, philosophically, I'm assuming it's in stasis, and I'm not really seeing degradation, although it does happen, and you will see changes. We talk about container closure, gas ingress, but you still get some molecular mobility depending on your formulation. But freeze-thaw studies are one of the first things I do with these sorts of products to understand getting in and out of state. It's often a critical point. And when we talk about testing and the earlier question about time to analysis, really make sure that your analytical testing and your stability testing is structured in a way that you're not creating artifacts from the test procedure, which is often you'll add damage to damage. And so in your mRNA is not mRNA, and we understand very little about secondary structure about mRNA and formulation development. We're understanding more and more. Biologics, I'm going to say, are relatively simple in some cases because the amount of work that's been in traditional proteins, but still extremely complex. And in my mind, there's no substitute for real-time stability data. So for early stage programs, enroll early, You can usually allow for the regulators to accept a real-time plus argument, 6 months, and I've done up to 12 months. So filing based on 6 months. And lead stability is critical. So as you're going into your clinical programs, leverage your early development lots, leverage your engineering runs, get those in stability as early as possible, recognizing that container closure and formulation differences can impact the final drug product stability for your clinic. So if you've made changes, there's always going to be an asterisk, but that's usually how you get into these early stages. And the batch end, in my experience, is often extremely low, so you don't have the power of statistics to look at replicates either. So hopefully that helps.
Very much. Thank you so much. Appreciate it.
Okay, thank you. We're just a little ways past the halfway point, so it's time for a commercial here. I'll take this opportunity to invite those who are new to Stability Hub, those of you who come through us through LinkedIn invitations, to visit our website, stabilityhub.com, check out our featured publications, regulations, events, suppliers, interactive forum, job opportunities, and more. And while there, sign up for our monthly newsletter to see what's new. And then we're always looking for people to participate with us in this vast stability world. If you serve on an industry committee, such as the ICH Revision Committee or anything else along statistics lines or anything like that, we're glad to have you as our representative there. You're our volunteer representative. You all get the same pay that I do, and to report back so we can share that kind of information with the rest of the world. So that's greatly appreciated. There's also archival opportunities, those kind of things. We're always glad to have any of your summer interns that you find sitting at their desks with nothing to do. You can teach them a lot about stability by having them combing our site for some of the research opportunities that we have. And then as part of the Pharmaceutical Stability Discussion Group, or PSDG for short, Stability Hub is exploring returning to live meetings. If you've enjoyed this format, it is so much easier and more enjoyable to actually be in the same room with everybody else. But circumstances have kind of taken that away in recent years, and we're looking to overcome that and get back to it. In the live PSDG meetings, attenders can spend parts of 3 days asking questions, sharing information, touring facilities, meeting vendors and their services, and networking to gain lifelong professional contacts and friends. There could be a meeting as early as October or no later than next May as we're doing our explorations. We're looking for companies in the US that would like to host a meeting at their facilities. If you'd like to learn more about attending or hosting such a live meeting, send an email to john@stabilityhub.com, and we'll be glad to give you more information and see what we can do. But we really hope to return to live meetings at some point in addition to these webinars. We find that to be very useful. End of commercial. Let's talk about stability chambers for just a little bit. And wanted to ask Jeff Cook about the potential use of AI and some of the new developments when it comes to stability chambers and what you're thinking about over parameter and generation control.
Certainly. Thank you, John. Yes, so the AI is, I'm incredibly excited about. As the air handler manufacturer, my focus is a bit less on the data and more on the performance of the machine. And AI will actually help both of those. I'm looking at redundancy seems to be a big buzzword around the industry these days, and defining what redundancy actually is seems to be a big topic. But I'm hoping the redundancy will be helped by the AI sort of through auto-tuning functions of the AI being able to look at the performance of the machine and notice patterns of performance and optimize that performance. Talking, auto-tuning PID values based on the ambient changes in ambient environment in relation to the internal stability chamber conditions. Accelerated ramping with stable dwells, we would look to have auto-tuned PID values to increase the ramp rates while also maintaining stability at your dwells. And then with redundancy, we're talking auto-tuned not only PID values, but also transition rates during unit switchover, either if it's on a 4075 that's switching over on a time-based approach, or if it's a freezer that switches when it inevitably freezes. So we have the AI sort of looking at the performance data in relation to the advanced settings of the machine and crunching all of that data to achieve a seamless transition during redundancy switchover. So the machines can actually switch between the two, but the data that you see from the temperature and humidity side is seamless. There is no defrost spike. There is no switchover lull in performance if you're going from a 4075 unit that has encountered a fault or is undergoing maintenance. That is all internal to the machine. Really excited about the AI functions there, but also, you know, for the users in terms of water usage based on the humidity levels achieved inside the stability chamber and how much water we use in relation to the ambient conditions. I think we will also be able to build power efficiency into the refrigeration systems. The new refrigeration components, and even beyond the refrigeration components, the fans and everything, they're all being increasingly built with feedback loops to provide active data. And what we can do is then we can crunch that data to have the fan speeds modulate along with variable speed compressors and electronic valves that have their own microprocessors. So the whole refrigeration system is quote unquote smart. And rather than everything sort of running all out and the heaters fighting the refrigeration system, they will be actively talking to each other and working together to better the energy usage of the machine. Beyond that, obviously the data analytics comes into play. Parameter's new controller offers about 35 data variables, and they are taking readings every 10 seconds. And so that amount of data adds up to almost an insurmountable amount of data for any person to be able to crunch into something constructive. So the software and the AI will be able to find patterns in the data, maybe be able to identify patterns of unit performance and sort of learn how to make the machine perform optimally based on that data.
I find it real interesting. You mentioned the war between the various factions. I remember having that explained to me years ago, that it was a war between the heaters and the coolers and the dehumidifiers and the humidifiers. And whenever one went down or weakened a little bit, then everything shifted, went right off the table. So it's nice to know that they could cooperate with each other and sort of meet in the middle.
Exactly, John, exactly. You know, depending on the theory of operation and how you're attaining your humidity, you know, you're going to inject a little bit of steam and then you're going to fight that steam with some cool air. Or in the parameters dew point method, you do have a refrigeration compressor and then also an air heater. And, you know, as you go up and down around your set point, those two components are, in a sense, fighting each other. So the ability of them to talk to each other, I'm looking at greatly increasing the power efficiency of the machine.
Thanks. I know you guys are the next up in the webinar series for talking about validation, calibration, that kind of thing. One of the questions that we get almost every time we meet is how often you need to validate a stability chamber, requalify a chamber. And typically comes up in our polls that people are doing it anywhere between 1 and 3 years, with a few folks as necessary on the outside. And that's always a fallback, as necessary means whenever you make some kind of a change to your load or unlike equipment, change out, that kind of thing. But it's interesting. We'll talk more about that when we get to that particular webinar. But if anybody has any questions about how often it is, and it's an urgent question, we'd be glad to speak a little bit about it now.
Would somebody be able to explain that? Because that's one of the things that we get on audits. a lot, and we really don't know the exact answer and like how to kind of like what's the like perfect time to do that, I guess.
Sure, it's kind of depends. Again, it's one of those depending questions. Some companies, just to avoid a problem on an audit, will do it every single year. And that's expensive to do, but it does take that club out of the hand of the auditor. The other way to go is to point to some guidelines. If you look at the WHO guidance, it says every 2 to 3 years. And someplace or other I went searching for when I found that WHO guidance, I had seen that it was required every 2 years, but it wasn't an FDA guidance. I think it was something international or maybe over in Europe that they talked about every 2 years. And then industry, when you take a poll, a lot of them will say, well, 3 years is a good time to double back and check on it. So you'll find all those 3 times done that way. And then a few people that just say, you know, from time to time we have changes, we're going to have to do it anyhow there. So let's just make our point wherever we make a change. If no change occurs, then we don't have to do anything. So all of those answers are acceptable as long as you can justify them and point to that, because it is all over the map so far. But I would say that when your auditors ask you that, just to have great justification as to here's what industry typically does, here's what a guideline says, here's our data that shows everything's been running okay. All those things can go into a justification. Anybody else want to comment? Bashar, your square is lit up. Did you want to comment?
Oh, I just had another quick question. So we've gotten away with it a lot from our biologics side, but it's really more the pharmaceutical people that are very like nitpicky about it. Because we always use the excuse that if engineering approves it we do like an annual review of all our chambers at the end of the year.
Mm-hmm.
And if engineering sees that there was no issues with them for the year, we never revalidated them because we use that as our thing. But the pharmaceutical people found that still to be like not acceptable. So now we're trying to figure out what the best timeline is for that.
Okay, just as a couple of references, I found the WHO Annex 9 titled Temperature Mapping of Storage Areas. And associated with that is the technical supplement 961 of the same title.
Okay. Cool. Thank you.
Yeah, I'm sure the ISPE folks have great recommendations. They have a great guidance that is already written into the article we'll be talking about for the next month's topic. But it's expensive. It's over, I think, $900 or something to get a copy. And if I was working for a large pharmaceutical corporation like I had been, I would simply put in a purchase requisition and they would get it for me. So I've not had access directly into one of those. But if anybody wants to buy one and share it with us, we'd be glad to spread the knowledge around. But ISPE has been great when it comes to stability chambers and trying to put some guidances together. They're not an official requirement, but they're as close as you can get for knowledgeable people coming up with very realistic kind of suggestions.
Cool.
Anything else in the chamber area? Okay, we will check our chats. Don't see anything new at the moment. Let's talk about significant changes when it comes to our stability data. If we're going along nicely and suddenly we get a significant change, we had a question, or at least a point made, if a significant change is observed in an accelerated condition sample, in a stability sample for a zone 4A territory, example, a degradation product that exceeds the specification limit, does that mean that no shelf life extrapolation will be accepted as the zone 4A real-time storage condition is the same as the condition generally applied as intermediates, as per Q1E, which is getting replaced by some of the other ones. So any comment on that? What your experience with significant changes? Generally, if it's something at an accelerated temperature and it's not a zone 4 condition, let's say it's zone 2 or 3, then you would fall back to a 30-degree point from your 40 and then continue on with that particular one. Jeff? Your hand is raised.
Yeah, I've unmuted it this time, so I hope that makes a difference.
It's working.
Yeah, it
Although now
worked.
you're speaking in a British accent.
Oh, yeah, well, that's where I am now. Yeah, that's right. So typically you're allowed to do some shelf life extrapolations as per ICH Q1E. The extrapolation is greater if you have no significant changes under your accelerated conditions. But if you do get a change, then you're supposed to revert to your intermediate sample conditions. And if those then are satisfactory, you can still do some extrapolation, but not as much as you could if you had got no significant changes under accelerated conditions. But now, if you're working for a zone 4A or zone 4B territory, your long-term condition, your real-time condition, is the same as that intermediate condition. And so now you don't have an intermediate condition anymore. You only have the accelerated condition. So it seems to me that that really means that you're just not going to be allowed to have any shelf life extrapolation at all, but just wanted to check whether others have had any experience with this.
It's a good question. I hope people are thumbing through the files of their minds, thinking back to any time that situation might have occurred.
It also goes back a few years when ICH initially brought in this idea of the intermediate condition, because it was originally brought in, as I understood it, to help third world countries get new drug materials into their place, because there was a reluctance to do stability under third world type environmental conditions, because it would add expenses to the drug development. So ICH introduced this concept of an intermediate condition, which would enable companies to generate data for third world countries more cheaply, or something like that. But really, I think a lot of companies then decided, well, we're not going to do this because it means that if we don't get clean results under our accelerated conditions, we won't be allowed to do any shelf life extrapolation. So this would be a blockage to operating in that way.
Okay, thank you. You mentioned ICH, and the big elephant in the stability chamber rooms are the big revision that we have for the ICH stability guidances. There are several included there, including the Q5A for biologics. And I was wondering, has anybody had a chance to kind of dive in there and take a look? Any concerns that you saw or observations you made about that? I started on it. It's out for comment, and in that format, every single line has a number and is spread apart. I found that hard to read, so I decided I would just take it on myself to make those into paragraphs that made sense to me, and take the numbers out, and that slowed me down so much. I've gotten to all the introduction stuff, and so all the reasons why they wanted to do that and where they made the changes, but I didn't get to see the changes themselves yet. So anybody else want to comment on the new guidances coming, or new guidance coming through? Maria?
I was just very curious if my company has been looking at it a lot because it addresses accelerated stability, but if anyone knows what the end of the comment period is, because they were released on April 11th, but we haven't been able to find a date on the website.
I look for that, but I'm guessing if you find it under the original release in the CFR or under thing, it probably would tell you what the end of the comment period is, but maybe somebody's already seen that. Nobody's seen it yet? Okay, or have you done any reading in it that brought you some information?
mean, I would say for anyone who's interested, you know, there was a question about sort of where Arrhenius modeling and other types of modeling are appropriate for biologics. That's definitely something that's discussed in it. I know my company has some feedback on it, but you know, there is a lot more robust discussion of kind of other modeling approaches.
I was glad to hear that they're doing the kind of a revamp of it, but my philosophy has always been not to do huge documents. So wherever you make a small revision, you got to go back and everybody has to retrain and reread everything. I'm kind of a fan of the smaller ones. If you look at the government process of big, beautiful bills and things that are a zillion pages long so that nobody actually gets to read it before it gets passed, I'm kind of concerned that that'll be the case for this document. But be that as it may, it's the latest thing in stability. It's got some nice new updates, and we'll learn to love it, I guess, especially once all the comments come back in. Okay, nothing else on the guidance. Another question, can water activity be used to determine storage and stability conditions for lyophilized products? So I'm guessing that that would have a significant impact on it, but I've not done a lyophilized product for a number of years. Can anybody else comment on that? Especially if you've done a stability study on a cake that you thought was mostly powder and you tried to break it up a little bit and discovered it was a piece of concrete. Give you a second to think about that. So maybe no lyophilized folks online at the moment.
I guess question, when you say in terms of determining storage and stability, are you talking in terms of the water content requirements or impact on stability? Because we've definitely done that.
Jeff, that was your question. Do you want to unmute and ask it a little bit more in detail?
Sorry, yeah, I was just trying to get to my unmute button. Yeah, so I'm just wondering, and I thought Ken might jump in there. You know, how can water activity affect stability conditions for lyophilized products, and you know, and what would be the best way to kind of measure for that water activity?
Sometimes at PSDG meetings, we're so generalized that we don't get to answer some of the very specific questions, but
I mean,
they certainly resound with us, and something we can take back to our colleagues and start asking around, particularly if that's their specialty.
Yeah, I just wondered if Ken Victor was still online.
Yeah, I'm online. I'm not certain about exactly what you mean by the question. I mean, I think the idea is that I think what is trying to be conveyed is that the water activity of the lyophilized product being related to the stability of it, and can you use water activity measurements instead of alternatives like Carl Fisher assays to measure the water content. I don't know if that's essentially
Yeah,
what
that was the kind of question that I meant. Yeah.
Well, so one thing to consider, water content is obviously, as it says, it's the percent water content in the product. But that doesn't necessarily mean that water content is available for microbiological degradation, versus water activity of the product measures for the free water particles available in the product, which is actually available for and affects the microbiological degradation. So there is a difference between water content and water activity. Specifically to your question, water activity is obviously very normally used in small molecules, especially for information only testing, kind of to understand what's going on with your product. I haven't necessarily seen that being used for lyo products. We have a lyo vaccine that's on stability right now as we speak, and we don't use water activity. But again, if that answers your question, water activity, it basically talks to how much of free water is available in your product, and that is directly affects increasing the micro content and degradation.
So
Okay,
when you
thanks.
stated, oh, sorry.
No, Congan.
So when you stated that you do not currently use water activity measurements to evaluate product stability, but you acknowledge that that is a better metric than water content, is there a particular rationale right now why you are not using water activity?
Well, I can't comment on if it is a better metric, but in small molecules world, right? So especially when there is not a lot of information available on the product and you want to understand if there is increase in the micro content, TMC, TYMC, and you want to get a preliminary read and to understand, then you go ahead and do the water activity. Water content is more about your active plus excipients together would have an expected level of water content that you think is inherent to the product. Usually it is less than 5%. And you measure over a period of time if that's changing or not. So if you have a problem with water activity, I would, in my understanding, your water content, you're actually expecting some activity there to go up. If that makes sense. So.
Maria?
I could chime in. So I agree so much with that answer about the water activity versus water content, sort of giving different information. I have a strange perspective. I don't have a great sense of how, from the regulatory sense, those two are compared. But for our models, we need to relate a water measurement to chemical behavior. And I would say my experience has been the water activity tends to be a much more consistent measurement that's less affected by, say, lab conditions or speed of exchange with the compound. Then say a lot of the standard in pharmaceutical is KF, and there's a lot of very questionable KF measurements. So having seen them from a product behavior point of view, I actually think it's a much more robust measurement. Don't know from the regulatory sense.
When there is a USP 922 document that focuses on water activity, but it's the way it was written, it tends to focus more on oral solid dosage type product as opposed to lyophilized product. And that's kind of, I guess, the question a little bit is why in the oral solid dosage and in the food industry, they recognize water activity as a more important metric of product stability, or while in the lyophilized world, it's really been focused, I guess it's probably historically, on the moisture content instead, and therefore focused on using things like Carl Fisher titration. I don't know if anyone has any comments about that, but it would be nice to see the industry moving towards using water activity measurements as a more direct relation towards stability instead of the moisture content as measured by Carl Fisher. But I don't know what has to happen in order for that to occur.
Okay, Jeff, your hand was raised.
As was said, there is a USP monograph 922 I was a little bit involved with that particular monograph. And when I was with the General Chapters Committee, and it was done in conjunction with the microbiological group. As Sri was saying, it's related to microbiological growth. So the idea here is that microbiological testing of solid oral dosage forms is required. It's required for release testing, for stability testing. But the idea here is that if the water activity within the product is below a certain figure, and I believe that's something like about 0.5, then microbiological growth can't happen. And so you don't have to do the microbiological testing. And I think that's the concept that was being used in the USP and the rationale behind bringing that in as an alternative. It is quite a different determination than Carl-Fisher determinations or loss on drying or whatever you want to do. I think that if you want to avoid using microbiological testing, you probably need to use both determinations. You need to know what the water content is of your product. So KF testing should be done. But then you want to know, well, there might be that much water in there, but is it available? And this is where the water activity determination comes in.
Okay, thank you. We've not done too many of the administrative kind of questions. We have just a few moments left. One of the ones that we had in advance was for early stage development, when should an organization start thinking about stability enrollment? When they would first do the studies, but early stage, in particular, I don't know about the therapeutics that was mentioned here, but typically R&D does a lot of the quick and dirty studies, and that would be especially true of some of the FreeThink involvements as well. And that it may not even be a formal stability program, but it certainly informs the later part of the stability. We like to think of product development reports and characterizing the product early on with a few of those early stability studies that eventually transition into the more formal ones for registration batches. So I don't know if people want to comment on that briefly as to when they think of their stability study program as taking off.
Yeah, I can jump in here. I mentioned earlier, never have enough stability from a CMC standpoint, and want to get a first look, especially for talking about biologics and cell and gene therapy products that are more complex, and you might not have the modeling tools for predictive stability to get those enrolled as early as possible. The challenges I've seen are on two ends, actually, at early companies over the last decade or so, is the worry that one, that these studies need to be full. They're not going to be QC. They're not going to be run by quality. So therefore that data is not valuable. And I don't believe that's true. I think the data is valuable. And the other end is where they're not controlled well enough, not documented well enough in early development or research, where the data becomes not useful. So I think you're always balancing those two items to make sure that the stability studies are appropriate. They don't have to be full ICH. They are often lead stability to help you understand your product and get an idea for which assays are going to be stability indicating, and it gives you a lead on starting to develop your formulation further and on matching up your assays to your needs there very early in development, because often that just takes a lot of time and material. But as I said earlier, you can never have enough stability. So I'm always a fan of over enrolling. It's easier to take it off stability later than to say, I wish I would have enrolled this lot. So when we're looking for production quantities, one of the big misses is the development teams or early manufacturing teams often don't account for the amount of material that is taken up by stability studies. That's just absolutely critical for product understanding.
Great, thank you. We've reached our expiry date time-wise here, so we'll ask anyone that remaining questions or anything that pops up in the future. You're welcome to put them on the forum and Stability Hub. There's a great discussion forum there. We try to monitor that, and anybody who would like to respond to those kind of questions is welcome to. And as a staff, we'll try to do that ourselves. I wanted to thank Chris Latos for posting a link that we could get to a previous ISPE mapping paper. That would be of great value. And I'd like to close with a few words about the upcoming programs. So you can join us again on June 26th with a webinar by Parameter Generation Control on the validation, calibration, requalification, and mapping of stability chambers. There'll be an additional digitalization and chamber topics in our summer fall schedule. And the next Stability Hub office hours event will occur in October. Watch for announcements and opportunities to sign up with links in the Stability Hub newsletter and on the stabilityhub.com website as well. I want to thank our panelists for their valuable input to our program today, and you, the worldwide keepers of the stability function, shelf life of products, and safety of patients and consumers for attending. We trust that you found the information useful, and we hope to see you again soon. And with that, we'll bid you a good day. Thank you.
Although we need to end with the motto, the new motto from Warren: you can never have enough stability. Thank you, Warren, I love that.
Very good.
So noted. Closing words. Thank you.
Okay.
Bye all.
Bye.